Hello, friend. Welcome to Science for Sleep. Did you know that most vegetables are actually fruits? In a quiet botanical sense, anything that develops from the flower of a plant and carries seeds is a fruit. That means tomatoes, peppers, cucumbers, and even pumpkins belong to the fruit family. We call them vegetables in the kitchen because they are savory and often eaten with dinner. But in the slow logic of plant biology, they began as flowers. After a flower is pollinated, its base swells. The petals fall away. The ovary of the flower thickens. Inside, seeds begin to form. What we later slice and cook once held a blossom open to the sun. That small shift in perspective can feel gentle and surprising. The tomato resting on a cutting board was once a bright yellow flower. The pepper hanging from a stem was once part of a delicate bloom, designed to attract pollinators. A vegetable, in many cases, is simply a fruit that we decided to treat differently. Plants do not use the same categories we do. They are guided by structure and survival. If a plant part grows from a fertilized flower and contains seeds, it serves one purpose: to help the plant reproduce. The flesh around the seeds often becomes colorful as it ripens. Green fades. Red, orange, or yellow appears. This color change is a signal. It tells animals that the fruit is ready. When eaten, the seeds travel and may be deposited somewhere new. Some plants evolved clever ways to protect those seeds along the way. Peppers are one quiet example. The compound that makes a pepper feel spicy is called capsaicin. It creates a warm sensation in mammals. But birds are unaffected by it. Their bodies do not register the heat in the same way. Birds can eat peppers without discomfort, and they swallow the seeds whole. Later, those seeds pass through and may sprout in new soil. Mammals, on the other hand, tend to chew seeds, which can damage them. Over time, peppers developed a chemical defense that gently discouraged mammals, but allowed birds to help with seed spread. It is a small evolutionary adjustment, built through many seasons. Then there is another gentle surprise hiding in the garden. Broccoli, cabbage, kale, cauliflower, and Brussels sprouts all come from the same wild species. That species is called Brassica oleracea. Long ago, along the rocky coasts of the Mediterranean, this plant grew with loose leaves and small flower clusters. Humans began selecting certain traits. Larger leaves were favored in some regions, tighter leaf heads in others, enlarged flower buds in another place. Over hundreds and then thousands of years, careful seed saving shaped these variations into what we now see as separate vegetables. But at their core, they share a single origin. The differences between cabbage and broccoli are not accidents of nature. They are the result of patient observation. A farmer notices a plant with thicker leaves. Seeds from that plant are saved. In the next generation, the trait becomes slightly stronger. Over time, a loose plant becomes a tight head of cabbage. A small cluster of buds becomes a sturdy stalk of broccoli. It is not a sudden transformation. It is gradual, almost quiet. When we look at a grocery store shelf, we see many kinds of vegetables, but beneath the surface there are shared patterns. Flowers becoming fruit, leaves folding into heads, buds swelling before they open, plants responding to light, temperature, and the steady rhythm of the seasons. And if you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find their way to this calm corner of the world, one sleepy traveler at a time. The quiet continues here, just as it is. The word vegetable is something we created. It is a human grouping based on taste and tradition. Plants, meanwhile, continue their cycles without needing those labels. They grow toward light. They form flowers. They set seeds. Some parts swell underground. Others ripen in the open air. What we gather and prepare is only one phase in a much longer story. Tonight, we begin that story gently. Beneath the names we use, there is structure. There is chemistry. There is sunlight turned into matter. There is a quiet, steady history unfolding leaf by leaf. When we begin to look a little closer, the word vegetable starts to feel softer and more flexible. It is not a strict scientific term. It is a way we describe certain parts of plants that we have chosen to eat. And those parts can be very different from one another. Some vegetables are roots. A carrot is a root. It grows downward into the soil, thickening as it stores energy. Beneath the surface, its orange body slowly expands. That thick root is not meant for us. It is meant for the plant. Inside the carrot, sugars made in the leaves are sent downward and stored. If the plant survives the winter, those stored sugars help it grow again in spring. What looks like a simple vegetable is actually a survival system, built quietly underground. Other vegetables are stems. A potato, even though it grows below the soil, is not a root. It is a tuber, which is a thickened stem. If you look closely at a potato, you can see small dimples called eyes. Each eye is a bud. In the right conditions, those buds can sprout into green shoots. That is because a potato is a modified stem, resting below ground. It stores starch in swollen tissue. When the time is right, it sends new growth upward toward the light. Some vegetables are leaves. Lettuce and spinach are collections of leaves, layered and folded. Leaves are the main site of photosynthesis. They spread outward to capture sunlight. Inside each leaf are countless tiny cells filled with chloroplasts. These structures contain chlorophyll, the green pigment that absorbs light. When we eat leafy vegetables, we are eating the very part of the plant that gathers energy from the sun. Then there are vegetables that are flowers, though we rarely think of them that way. Broccoli is made of tightly packed flower buds. If left unharvested, those buds would open into small yellow blossoms. Cauliflower is similar, except the flower tissue grows in a dense, pale cluster. Artichokes are also flowers, harvested before they bloom. In these cases, we are eating a plant at a very specific stage, just before it begins its next phase of life. Onions and garlic are yet another structure. They are bulbs. A bulb is made of modified leaves that wrap tightly around each other, forming layers. If you slice an onion in half, you can see those layers in neat rings. Each ring represents a leaf base, storing nutrients. At the center of the bulb is a small growing point. If planted, it can send up a green shoot. Beneath the soil, bulbs store energy and weight. All of these parts, roots, stems, leaves, flowers, bulbs, serve different roles in the plant's life. Roots anchor the plant and absorb water and minerals. Stems support leaves and transport sugars. Leaves gather light and produce food. Flowers enable reproduction. Bulbs and tubers store energy for the future. Even fruits, like tomatoes and peppers, are part of this larger structure. They begin as flowers. After pollination, the base of the flower swells and forms a protective layer around the seeds. The flesh develops to attract animals. Seeds are carried away, and the cycle continues. Plants are built from repeating patterns. Water moves upward through tubes called xylem. Sugars move downward or outward through tubes called phloem. Cells divide and expand. Tissues specialize. A vegetable is not a single type of thing. It is a plant part, shaped by both biology and time. If you imagine a garden from below the soil, you would see roots branching and thickening. Some would remain thin and fibrous. Others would swell into storage organs. Above the soil, leaves would tilt toward the light. Buds would form, some opening into flowers, others remaining tightly closed until harvested. The diversity we see in vegetables is a reflection of plant design. Each structure evolved for a reason. Each serves a purpose in growth, survival, or reproduction. And what we gather from the earth is simply one chapter in a much longer biological story, unfolding slowly, in light and soil, season after season. Light touches a leaf long before it ever becomes a carrot, a cabbage, or a tomato. At first, it is only energy. Sunlight travels across space and reaches the thin surface of a green leaf. The leaf appears still, almost quiet. But inside it, something steady is happening. Tiny structures within the leaf cells, called chloroplasts, are absorbing light. Inside those chloroplasts is a pigment called chlorophyll. Chlorophyll absorbs mostly blue and red light from the sun, and it reflects green light back outward. That reflection is what makes leaves look green to us. What we see as color is simply light that was not used. The light that is absorbed becomes energy for a process called photosynthesis. In simple terms, photosynthesis allows a plant to take carbon dioxide from the air and water from the soil and combine them, using sunlight, to create sugar. The chemical equation is quiet but powerful. Carbon dioxide, water, and light energy become glucose, a simple sugar, and oxygen as a byproduct. The oxygen drifts back into the air. The sugar stays. That sugar is the beginning of everything we call a vegetable. Glucose is a small molecule, but plants link many glucose units together to form larger molecules. Some of it becomes cellulose, which forms strong cell walls and gives structure to stems and leaves. Some of it becomes starch, a storage form of energy. When you eat a potato or a grain of corn, you are eating stored sunlight that was converted into starch. Inside a carrot root, sugars travel downward from the leaves through the phloem, a network of tiny tubes. The carrot thickens slowly as those sugars accumulate. In a potato tuber, starch granules fill the swollen stem tissue underground. The plant has no awareness of a future harvest. It is simply storing energy for itself, preparing for a time when growth will resume. Even the crisp snap of lettuce depends on sunlight transformed into structure. The cellulose in the leaf walls, built from sugars made in photosynthesis, gives shape to each cell. Water fills those cells and presses outward against the cell walls, creating firmness. That internal water pressure is called turgor pressure. When a leaf wilts, it is not because the structure disappeared. It is because water has left the cells and the pressure has dropped. Light also influences how plants grow toward it. This gentle movement is called phototropism. When one side of a stem receives more light, growth hormones redistribute within the plant. Cells on the shaded side elongate slightly more, causing the stem to bend toward the light source. It is a slow and patient adjustment, often invisible unless watched over time. Below the soil, roots respond to gravity. This is called gravitropism. Root tips sense direction and grow downward, anchoring the plant and seeking water. Shoots, by contrast, grow upward, toward light. These responses are built into plant cells. They are not decisions, but reactions shaped by evolution. Within each leaf, small openings called stomata open and close throughout the day. Through these openings, carbon dioxide enters, oxygen exits, water vapor escapes. When the air is dry, plants may close their stomata to reduce water loss. When the air is moist and light is available, they open again. This quiet regulation balances water conservation with energy production. The sugars made during the day do not remain in the leaf where they were created. Through the phloem, they are transported to other parts of the plant. Some go to growing tips, some go to roots, some become fruits or seeds. In this way, sunlight collected in one place supports growth in another. If you imagine a field at midday, leaves angled slightly toward the sun, each one is functioning as a small solar panel. But instead of electricity, it produces carbohydrates. Instead of wires, it uses living tubes of xylem and phloem. Instead of metal and silicon, it uses water, air, and green pigment. Vegetables are the visible result of this quiet conversion. They are matter built from light. They are structure assembled from carbon drawn out of the air. They are energy stored in roots, stems, leaves, flowers, and fruits. And all of it begins with something as simple and constant as sunlight resting on a leaf, moving through chlorophyll and becoming substance over time. Some plants choose to rise tall above the soil. Others, just as quietly, grow downward. If you were to kneel beside a garden and gently brush away the top layer of soil, you would begin to see a second world. Thin white roots branch and divide. Some remain delicate, like threads. Others slowly swell and thicken. Beneath the surface, vegetables often take shape where the air is cool and the light does not reach. Many of the vegetables we eat are storage organs. They are not random shapes. They are carefully built reservoirs of energy. During the growing season, leaves gather sunlight and turn it into sugars. Those sugars travel through the plant's vascular system. Some are used immediately for growth. Others are sent downward and stored. A carrot is a storage root. Its scientific structure is called a taproot. A taproot grows straight down and becomes the main anchor for the plant. As sugars accumulate, the cells in that root enlarge. Starch and other carbohydrates fill the tissue. Over time, the root becomes thick and firm. The bright orange color comes from beta-carotene, a pigment that the plant produces and stores within the root cells. That storage is not meant for us. It is meant for the plant's future. Carrots are typically biennial plants. In their first year, they focus on building that thick root. In their second year, if left in the ground, they use the stored energy to send up a tall, flowering stalk. The sugars saved underground fuel that sudden upward growth. The plant blooms, produces seeds, and completes its life cycle. What we harvest in the first year is a pause stage of that process. Potatoes follow a similar logic, but with a different structure. A potato is a tuber, which is a swollen underground stem. It forms at the end of horizontal stems called stolons. These stolons grow outward beneath the soil. At certain points, the tips begin to enlarge. Cells multiply. Starch accumulates. The tuber thickens. If you look closely at a potato, you can see small clusters of buds called eyes. Each eye is capable of producing a new shoot. That is because the potato is not a root. It is a stem with nodes, just like the stem above ground. When planted, those eyes awaken and send green growth toward the surface. The soil itself provides a steady environment for these storage organs. Underground temperatures change more slowly than air temperatures. Soil holds moisture and protects delicate tissues from wind and sun. For a plant preparing to survive winter, the earth becomes a quiet shelter. Onions and garlic also rely on storage, though in a different form. Their bulbs are made of tightly packed, modified leaves. Each layer stores nutrients and water. When conditions are right, the bulb uses those reserves to grow a new shoot. The layers we peel away are the plant's careful preparation for regrowth. Storage organs are shaped by the need for survival. In climates with cold winters or dry seasons, plants that could store energy had an advantage. When leaves died back or growth slowed, the stored carbohydrates remained safe below ground. When spring returned, those reserves fueled rapid new growth. Even the texture of these vegetables reflects their function. The dense crunch of a carrot comes from tightly packed cells filled with stored sugars and water. The firmness of a potato comes from starch granules held within its cells. When heated, those starch granules absorb water and swell, which is why cooked potatoes become soft and fluffy. Underground growth is not passive. Roots respond to gravity through a process called gravitropism. Specialized cells at the root tip sense direction. Tiny particles within those cells settle downward, helping the root determine which way is down. The root elongates in that direction, anchoring the plant and searching for water and minerals. Above ground, we see leaves and stems moving toward light. Below ground, roots move toward stability and moisture. Both directions are guided by built-in responses shaped by long periods of adaptation. If you imagine a field in early autumn, you might see only green tops swaying in the breeze. But beneath each plant, quiet structures are forming. Energy collected from the sun is being hidden away in the soil. Tissues are thickening. Starch is settling into cells. The plant is preparing for a season it cannot see, but is built to expect. What we harvest from the ground is not simply a vegetable. It is stored time. It is sunlight gathered months ago and preserved beneath the surface, waiting patiently for the next stage of growth. Before fields were arranged in rows, before tools shaped soil into neat lines, humans walked through landscapes that were already full of plants. In those earlier times, people gathered what grew naturally around them. Wild roots, tender leaves, seeds, and fruits were part of daily life. There were no grocery stores, no labeled categories. There were only seasons, weather patterns, and careful observation. Some plants were bitter, some were sweet. Some could be eaten raw. Others required roasting or soaking. Knowledge was built slowly, passed from one generation to the next. Archaeologists have found evidence of plant use stretching back tens of thousands of years. Tiny plant fibers have been discovered embedded in ancient stone tools. Seeds and starch grains have been found preserved in soil layers near old campfires. These traces suggest that early humans did not rely only on meat. They also gathered and processed plant foods, including wild ancestors of vegetables. Many wild plants were smaller and tougher than the vegetables we know today. Wild carrots, for example, had thin, pale roots and a strong flavor. Wild cabbage grew along rocky coastlines, with loose leaves spread outward. Wild peas were smaller and contained fewer seeds. But even in their early forms, these plants provided nourishment. Gathering required attention. People learned when certain roots were safest to dig. They learned which leaves were tender in spring and which became fibrous in summer. They learned how to avoid toxic plants that resembled edible ones. This knowledge was built through repetition, observation, and memory. Over time, something subtle began to change. Instead of only gathering plants, people began to influence them. If a root tasted better or was slightly larger, seeds from that plant might be saved and planted nearby. If a plant grew well in a certain patch of soil, that area might be cleared to encourage more growth. These small actions were the early steps toward cultivation. The shift from gathering to farming did not happen all at once. It unfolded gradually in different parts of the world. In the Fertile Crescent of the Middle East, people began cultivating grains and legumes around 10,000 years ago. In the Andes of South America, communities began domesticating potatoes. In East Asia, leafy greens and root vegetables were tended and selected. Each region developed its own relationship with local plants. As cultivation increased, people began to stay in one place for longer periods. Storing harvested crops made this possible. Roots and tubers, with their natural ability to store energy, were especially useful. A harvested potato could last for weeks or months in cool conditions. Onions and garlic, with their layered bulbs, could be dried and preserved. These storage qualities supported the development of settled communities. Farming also allowed people to shape plant traits more deliberately. Over generations, selective planting led to larger roots, tighter heads of cabbage, sweeter leaves, and less bitterness. Plants that once relied entirely on natural dispersal became connected to human care. Seeds were saved and transported along trade routes. Vegetables spread across continents through migration and exchange. The balance between plants and people became mutual. Humans provided protection, cleared land, and selected seeds. Plants provided food, stability, and seasonal rhythm. Fields replaced open gathering grounds. Irrigation channels replaced reliance on rainfall alone. Yet beneath these changes, the biological processes of plants remained the same. Leaves still gathered light, roots still searched for water, flowers still produced seeds, If you imagine an early cultivated plot, you might see uneven rows, soil turned by hand tools, and plants growing close together. There would be no machinery, only steady labor and careful timing. Seeds placed into soil in spring, shoots emerging weeks later, harvest in late summer, storage through winter. The vegetables we know today are shaped by this long partnership. They carry the history of wild ancestors and the imprint of human selection. Their forms reflect adaptation to climate, soil, and preference. Their diversity tells a story of movement across landscapes and cultures. And through it all, the cycle remains steady. Planting, growing, harvesting, saving seeds, beginning again. What started as quiet gathering became cultivation. What began as wild growth became tended fields. Yet beneath the surface of history, vegetables continue their ancient work, turning sunlight into matter, storing energy in roots and stems, and unfolding leaf by leaf in the rhythm of the seasons. For thousands of years, vegetables have been changing. Not quickly, not dramatically, but slowly, through a quiet and repeating pattern. The process begins with observation. A farmer walks through a field. One carrot is slightly thicker. One cabbage head is tighter. One wild lettuce plant tastes less bitter than the others. These small differences matter. Plants naturally vary from one another because of tiny changes in their genetic code. No two seeds are exactly the same. When a person notices a desirable trait, the next step is selection. Seeds from that particular plant are saved. In the next growing season, those seeds are planted again. Among the new generation, some plants will show the same trait. Perhaps the root is a little larger. Perhaps the leaves are softer. The cycle repeats. Observation, selection, saving seeds, planting again. Over many generations, this steady process reshapes a plant. Wild plants often carry strong defenses. Bitterness is one of them. Bitter flavors usually come from chemical compounds that discourage animals from eating the plant. Some wild carrots were woody and sharp in flavor. Wild cabbage leaves were tougher and more fibrous. Wild lettuce contained more latex-like sap. As people continued selecting plants that tasted milder or sweeter, those defensive traits slowly reduced. The plants that were easier to eat were planted more often. Over time, their chemical defenses became less intense. Size also changed. A root that stored slightly more sugar would grow thicker. A cabbage plant with leaves that wrapped more tightly around each other would form a denser head. Seeds from those plants were saved again and again. The differences accumulated. This process did not require knowledge of genetics. It relied on repetition. It relied on memory. It relied on paying attention. But there is another quiet effect of domestication. As selection focuses on certain traits, genetic diversity narrows, while plant populations usually contain a wide range of genetic variation. When humans repeatedly choose seeds from only a few plants with specific qualities, the genetic pool becomes smaller. The plant becomes more uniform. That uniformity can make vegetables predictable in size and taste, but it can also reduce natural resistance to pests or disease. Domesticated vegetables are shaped by preference. They are also shaped by movement. trade routes carried seeds far beyond their original regions. A vegetable that began in Central Asia could travel west into Europe or east into China. Spanish explorers carried tomato seeds from the Americas to Europe in the 1500s. From there, tomatoes spread across the Mediterranean and into Asia. Potatoes traveled similar paths, moving from the Andes into Europe and beyond. Each new environment influenced further selection. A plant growing in a cooler climate might be chosen for cold tolerance. A plant in a dry region might be selected for drought resistance. Over time, the same species could develop many regional forms. Domestication is not a single event. It is a loop. A farmer plants seeds in spring. The plants grow under sunlight. The best specimens are harvested, and their seeds are saved. The cycle begins again the following year. With each repetition, the plant shifts slightly. If you imagine this process stretched across centuries, you can see how dramatic change becomes possible without any sudden transformation. A wild plant with small leaves can, through thousands of careful selections, become a tightly wrapped cabbage. A thin root can become a thick carrot. A loose cluster of flower buds can become broccoli. The vegetables we know today are the result of this slow loop. They carry traces of their wild ancestors. They also carry the imprint of human hands choosing, year after year, which seeds to plant next. And now, as we begin to look at individual vegetables, we can trace these quiet changes more closely. Each one has its own origin story, shaped by geography, climate, and patient selection over time. If you hold a carrot in your hand, it seems simple. A smooth orange root, a narrow tip, a faint sweetness when you bite into it. But beneath that simplicity lies a long and quiet transformation. The carrot we know today, Daucus carota subspecies sativus, began far from modern farms. Its wild ancestors grew in regions of Central Asia, particularly in areas that are now Afghanistan and neighboring countries. These early carrots did not look like the bright orange roots we see today. They were often thin, pale, and sometimes purple or white. Their texture was firmer. Their flavor was sharper. Wild carrots grew in open fields and along rocky slopes. Their roots were smaller because their main purpose was not to feed people. The root stored energy so the plant could survive winter and send up a tall flowering stalk the following year. Carrots are naturally biennial. In the first year, they grow leaves and build a storage root. In the second year, if left in the soil, they use the stored sugars to produce flowers and seeds. That storage root is the part we harvest. Over many centuries, farmers began selecting carrots with thicker roots. A slightly larger root meant more stored sugar. A sweeter flavor meant a more pleasant taste. Seeds from those plants were saved and planted again. Gradually, the carrot changed. Purple carrots were once common in parts of Asia and the Middle East. These purple roots contained pigments called anthocyanins, which give plants deep red or purple colors. White and yellow carrots also existed. The now familiar orange carrot appeared later, likely in Europe around the 16th or 17th century. Dutch growers are often credited with developing orange varieties through careful breeding. The orange color comes from beta-carotene. Beta-carotene is a pigment that plants produce as part of their internal chemistry. It gives carrots their bright hue and also serves an important function for humans. When we eat beta-carotene, our bodies can convert it into vitamin A. Vitamin A supports vision, especially in low light, and helps maintain healthy skin and immune function. Inside the carrot root, beta-carotene is stored within plant cells along with starches and sugars. As the carrot grows, cells divide and expand. Sugars produced in the leaves travel downward through the phloem and accumulate in the root. The taproot thickens as these carbohydrates build up. Each cell becomes a small storage chamber, holding energy that the plant would otherwise use later. If you look at a carrot slice under magnification, you would see a central core and surrounding layers. These layers represent different types of tissue, including vascular tissue that once transported nutrients. The pattern is subtle but structured, shaped by the plant's growth. The soil environment plays a role as well. Carrots grow best in loose, well-drained soil. Compact soil can cause the root to fork or twist as it encounters resistance. In sandy soil, the root can grow straight and long. The shape of a carrot reflects not only genetics, but also the texture of the earth around it. Carrots are also sensitive to temperature. Cooler weather often enhances sweetness because lower temperatures encourage the plant to convert some starch into sugar as protection against frost. This is why carrots harvested after a light frost can taste slightly sweeter. Above the soil, carrot leaves gather sunlight through photosynthesis. Below the soil, the root continues to thicken quietly. If left unharvested into the second year, the stored sugars would fuel rapid upward growth. A tall, branching stalk would rise. Small white flowers would form in clusters called umbels. Seeds would follow, ready to begin the cycle again. The carrot's journey from thin wild root to thick orange vegetable was not sudden. It was shaped by patient selection across generations. Farmers noticed thicker roots. They saved those seeds. They planted them again. Over time, variation narrowed, and the carrot became more uniform in size and color. Yet beneath its cultivated form, the carrot remains what it has always been, a storage root, built to hold sunlight in solid form. It is energy preserved underground. It is sweetness gathered over a season and kept safe in the soil. And from this root beneath the surface, we can move even deeper into the ground, toward another vegetable that sleeps below the soil in a different way. Deeper beneath the soil, past the fine threads of roots and the steady tap of a carrot, there is another kind of storage organ resting in the dark. The potato does not grow as a root, it grows as a stem. The potato, Solanum tuberosum, began high in the Andes Mountains of South America. In regions that are now part of Peru and Bolivia, communities were cultivating potatoes thousands of years ago. The cool mountain air and rocky soil shaped its early growth. Unlike carrots, which store energy in a thickened root, potatoes store energy in a structure called a tuber. A tuber is a modified underground stem. Above ground, stems have nodes and buds. They branch and produce leaves. A potato tuber has those same features, but they are hidden beneath the soil. The small dimples scattered across its surface are called eyes. Each eye contains a bud. Given warmth and moisture, that bud can sprout into a new plant. If you cut a potato into pieces making sure each piece contains at least one eye, and place those pieces in soil. Each one can grow into a new potato plant. That is because the potato tuber is living stem tissue, resting and waiting. Potatoes form from underground stems called stolons. Stolons grow outward horizontally beneath the soil. At certain points along the stolon, cells begin to divide and swell. The tip thickens. Starch begins to accumulate. Over time, the swollen tip becomes a tuber. Inside a potato, most of the stored energy is starch. Starch is made of long chains of glucose molecules, which were originally produced through photosynthesis in the leaves. During the growing season, sugars travel from the leaves down through the phloem and into the developing tuber. There, enzymes convert the sugars into starch and pack them into tiny granules within the plant cells. The potato becomes a storage vault of sunlight. The cool, stable environment of the soil protects these tubers. Underground temperatures fluctuate more slowly than the air above. The soil shields the tuber from wind, light, and sudden frost. In the Andes, this protection was especially important. Harsh mountain conditions favored plants that could store energy safely beneath the surface. Like carrots, potatoes are also part of a survival strategy. The stored starch allows the plant to regrow if the above-ground leaves are damaged or if winter interrupts growth. When the next growing season arrives, the buds in the eyes begin to sprout. A new stem pushes upward. Leaves unfold. Photosynthesis resumes. The distinction between root and stem may seem small, but it shapes how the plant grows. Roots absorb water and minerals. Stems support leaves and transport nutrients. A potato structure allows it to regenerate from multiple buds, which made it especially valuable to early farmers. After the Spanish encountered potatoes in the 16th century, they carried them back to Europe. At first, the plant was met with caution, but over time, it became an important staple. Its ability to produce a large amount of food from a relatively small area of land made it reliable. The chemistry of the potato also shifts depending on how it is handled. When a cooked potato cools, some of its starch reorganizes into a form called resistant starch. Resistant starch is less easily digested in the small intestine and can travel to the large intestine, where it becomes food for beneficial gut bacteria. This transformation happens quietly as the potato cools, without any visible change. Potatoes must also be stored carefully. Exposure to light can cause the tuber to produce chlorophyll, turning parts of it green. Along with chlorophyll, the potato may produce compounds called glycoalkaloids, which can taste bitter. This is a natural defense mechanism. In the soil, light exposure is rare, but once harvested, light can trigger these changes. If you imagine a field at harvest time, the green foliage above ground may begin to yellow and fade. Beneath the surface, clusters of tubers rest in the soil. Each one formed from a hidden stem. Each one filled slowly with starch as sunlight moved downward through the plant. The potato sleeps underground as a stem disguised as a root. It stores energy. It waits. And from this quiet storage organ beneath the soil, we begin to move upward again, toward vegetables that hang in open air, ripening in color beneath the sun. Above the soil, hanging lightly from a vine, there is a vegetable that is not truly a vegetable at all. The tomato, Solanum lycopersicum, begins its life as a small yellow flower. Each blossom opens toward the sun, its petals spreading outward. Inside the flower, pollen moves from one structure to another, either by wind or by the vibration of visiting insects. When pollination occurs, something subtle begins to change. The petals fade and fall. The base of the flower starts to swell. Cells divide. Tissue thickens. The ovary of the flower becomes a fruit. Botanically, that swelling structure is a fruit because it develops from a fertilized flower and contains seeds. In the kitchen, we place tomatoes beside lettuce and onions. But in plant biology, they belong to the same category as berries and grapes. Tomatoes originated in Central and South America. Wild ancestors grew in regions of present-day Peru and Ecuador. Early forms were small, closer in size to cherry tomatoes. Indigenous communities began cultivating and selecting larger varieties long before the plant reached Europe. When Spanish explorers encountered tomatoes in the 16th century, seeds traveled across the Atlantic. From Spain, tomatoes spread into Italy and the Mediterranean, eventually becoming deeply rooted in regional cuisines. As a tomato ripens, its chemistry shifts in a visible way. When young, tomatoes are green. This green color comes from chlorophyll, the same pigment that allows leaves to perform photosynthesis. At this stage, the fruit is still contributing to the plant's energy production. As ripening begins, chlorophyll breaks down. At the same time, other pigments increase. One of these pigments is lycopene, which gives ripe tomatoes their red color. Lycopene belongs to a group of compounds called carotenoids. These pigments absorb certain wavelengths of light and reflect others, producing shades of red and orange. The transition from green to red is more than a change in color. It signals that the seeds inside are mature. The fruit softens. Sugars increase. Acidity adjusts. The tomato becomes more appealing to animals that might eat it and disperse the seeds. Inside the tomato, seeds are embedded in chambers filled with gel-like tissue. This moist environment protects the seeds as they develop. Once the fruit is eaten or decays on the ground, the seeds are released and may germinate if conditions are right. Tomato plants themselves can grow in two main patterns. Some varieties are determinate. Determinate plants grow to a certain height, set fruit over a relatively short period, and then stop. Others are indeterminate. Indeterminate plants continue growing and producing fruit throughout the season, extending their stems and forming new flowers as long as conditions remain favorable. This growth pattern shapes how tomatoes are cultivated. Determinate varieties are often used for large-scale harvesting because they produce fruit all at once. Indeterminate varieties may be supported by stakes or trellises, climbing upward and producing gradually over time. The tomato plant is part of the nightshade family, known scientifically as Solanaceae. This family includes potatoes, eggplants, and peppers. Some members of this family produce compounds that deter insects or animals. In tomatoes, small hairs on the stems and leaves can provide mild protection against pests. Temperature also influences tomato growth. Tomatoes prefer warm conditions. Cold temperatures can slow ripening or damage developing fruit. In cooler climates, gardeners may start seedlings indoors before transplanting them outside once frost has passed. If you imagine a tomato vine in late summer, heavy clusters of fruit hang at different stages of ripeness. Some are still green, some are pale orange, others are deep red, reflecting sunlight with a soft sheen. Each fruit represents a flower that once opened and then quietly transformed. Though we call it a vegetable, the tomato is a fruit built to carry seeds. It ripens in open air. It changes color as pigments shift within its cells. It softens as sugars rise and acids balance. Its journey moves upward toward light and warmth. From this bright and ripening fruit, we begin to turn toward another plant that grows close to the soil, forming tight layers beneath the surface, waiting patiently for its own season of growth. Near the surface of the soil, where roots meet air and stems begin to rise, there is a plant that grows in circles. The onion, Allium cepa, does not form a thick taproot like a carrot or a swollen stem like a potato. Instead, it builds a bulb. A bulb is made of tightly wrapped, modified leaves that store nutrients and water. If you slice an onion in half from top to bottom, you can see these layers clearly. Each ring is a leaf base pressed against the next. The onion's story likely began in regions of Southwest Asia, where wild relatives grew in dry, open landscapes. In these environments, rainfall could be unpredictable. Plants that could store water and energy in a compact structure had an advantage. The bulb allowed the plant to survive dry seasons and then regrow when conditions improved. Unlike many vegetables that respond mainly to temperature, onions are also sensitive to the length of daylight. This sensitivity is called photoperiodism. Onion plants begin forming bulbs when they receive a certain number of daylight hours. Some varieties are long day onions, meaning they require many hours of daylight before bulbing begins. Others are short day onions, forming bulbs when days are shorter. Inside the plant, cells detect changes in light duration through specialized pigments. These signals influence hormones that shift the plant's growth from leaf production to bulb formation. Leaves continue gathering sunlight, but energy begins moving downward into the expanding layers below. As the bulb develops, the bases of the leaves thicken. Sugars and nutrients accumulate between cell walls. Water fills the tissues. The bulb swells outward in smooth, rounded form. Underground, it grows quietly in concentric layers. Onions are biennial plants. In their first year, they form leaves and a bulb. If left in the soil for a second year, the stored energy in the bulb fuels rapid upward growth. A tall flower stalk emerges. At its top, forms a round cluster of small white or purple flowers. Seeds develop within these blossoms, completing the life cycle. The chemistry of the onion also carries its own quiet story. When you cut into an onion, you may notice your eyes begin to water. This reaction comes from sulfur-containing compounds stored within the bulb cells. When the onion is intact, these compounds remain separated inside different parts of the cell. But when the onion is sliced, the cell walls break. Enzymes mix with sulfur compounds, producing a gas that rises into the air. When that gas reaches the moisture in your eyes, it forms a mild acidic compound. The eyes respond by producing tears to dilute and wash away the irritant. This reaction is a defense mechanism built into the plant. Underground, sulfur compounds can discourage pests and protect the bulb from being eaten. After harvest, onions undergo another change. The outer layers of the bulb dry and become papery. These outer layers form a protective skin that helps reduce water loss. Properly cured onions can be stored for months in cool, dry conditions. During storage, the bulb remains alive, though growth is paused. If you imagine an onion field nearing harvest, green tubular leaves rise above the soil. As maturity approaches, the tops may bend and begin to dry. Beneath the soil, bulbs rest in the earth, their layered interiors complete. Each layer represents time. Each ring once gathered sunlight as part of a leaf. Those leaves thickened at their bases, storing energy for future growth. When we peel an onion, we move backward through those layers, touching the plant's stored reserves. The onion's growth is guided by light, shaped by day length, and protected by chemistry. It grows in cycles of storage and release, building structure beneath the surface before rising again toward the sun. And close beside the onion, sharing a similar shape and origin, there is another bulb that does not rely on seeds in the same way, but instead carries its future within separate, tightly packed segments. Close to the onion in shape and scent, there is another bulb that grows beneath the soil, wrapped in pale skin and divided into quiet segments. Garlic, Allium sativum, likely began in Central Asia. Wild relatives still grow in mountainous regions, where cold winters and dry summers shape their survival strategies. Like the onion, garlic forms a bulb made of modified leaf bases. But instead of building one solid round bulb, garlic divides itself into individual sections called cloves. Each clove is not just a fragment, it is a complete starting point. When a clove is planted in soil, it sends down roots and pushes up green shoots. It does not need seeds to reproduce. Instead, it relies on vegetative reproduction. This means new plants grow from parts of the parent plant, rather than from fertilized seeds. Because garlic is usually propagated by planting cloves rather than seeds, cultivated garlic has very little genetic variation compared to wild plants. Each new garlic plant is essentially a clone of the one before it. The same genetic pattern is carried forward season after season. This cloning through time has advantages and limits. It allows farmers to preserve specific traits, such as bulb size or flavor intensity. But it also means that cultivated garlic populations can be more vulnerable to disease if new threats arise. Without wide genetic diversity, adaptation can be slower. Garlic forms its bulb gradually as daylight increases. Like onions, garlic responds to photoperiod. As days grow longer, signals inside the plant shift its energy toward bulb development. Leaves continue capturing sunlight, and sugars move downward into the swelling cloves. Each clove develops around a central stem called the basal plate. Roots grow from the bottom of this plate into the soil. The outermost layers of the bulb dry into a thin, papery covering that protects the cloves during storage. One of garlic's most well-known traits appears when it is crushed or chopped. When intact, garlic cells keep certain sulfur-containing compounds separate from specific enzymes. When the cells are broken, these components mix. An enzyme called allinase converts a compound called alleine into allicin. Allicin is responsible for garlic's sharp aroma and much of its biological activity. It is short-lived but reactive. In plants, sulfur compounds help defend against insects and soil pathogens. In humans, allicin has been studied for its antimicrobial properties. It can interfere with certain bacteria and fungi, though its effects depend on concentration and preparation. Garlic's strong scent, then, is part of its defense system. If you look at a garlic bulb before planting, each clove is wrapped tightly in a thin skin. The bulb itself rests quietly, storing energy and water. When placed into soil, the clove awakens. Roots extend downward, guided by gravity. A shoot rises upward, guided by light. Leaves grow long and narrow, resembling flat blades. Garlic plants prefer well-drained soil and cool growing conditions during early development. In many regions, cloves are planted in autumn. They develop roots before winter, then pause during the cold months. In spring, growth resumes, fueled by the energy stored within the clove and later supported by photosynthesis. By summer, the plant begins forming a new bulb beneath the soil surface. The original clove that was planted has now divided into multiple new cloves. Each of those cloves contains the potential for another plant. If left unharvested into a second year, some garlic varieties may send up a flower stalk called a scape. At its tip, small bulbils can form instead of seeds. These bulbils are miniature cloves, another method of vegetative reproduction. Garlic's life cycle is steady and repetitive. Clove becomes plant. Plant forms bulb. Bulb divides into cloves. Cloves are planted again. It is a quiet lineage, carried forward by separation rather than pollination. Beneath the soil, garlic grows in clusters of pale segments pressed together in a circle. Each one stores sunlight gathered through green leaves above. Each one holds within it the instructions for another season of growth. From this pungent bulb with its tightly packed cloves, we begin to move upward again, toward vegetables whose energy is stored not in roots or bulbs, but in leaves that gather light openly and unfold toward the sky. If you hold a head of lettuce, it feels light. When you tear a leaf, it snaps softly. There is a quiet crispness to it, almost like breaking a thin sheet of ice. Lettuce, Lactuca sativa, was domesticated from a wild ancestor known as Lactuca cereola. Wild lettuce plants were taller and more upright, with thinner leaves and a stronger, slightly bitter taste. They grew in regions around the Mediterranean and parts of Western Asia. Over time, farmers selected plants with broader leaves, milder flavor, and softer texture. Unlike carrots or potatoes, lettuce does not store large amounts of starch underground. Most of what we eat is leaf tissue, and those leaves are mostly water. Fresh lettuce can be more than 90% water by weight. That high water content gives lettuce its cool and refreshing quality. But it also means the plant depends heavily on internal water pressure to maintain structure. Inside each lettuce leaf are millions of plant cells. Each cell has a rigid outer wall and a central compartment called a vacuole. The vacuole stores water and dissolves substances. When water fills the vacuole, it presses outward against the cell wall. This pressure is called turgor pressure. Turgor pressure keeps the leaf firm. When lettuce is freshly harvested and well hydrated, its cells are full. The pressure pushes outward evenly, giving the leaf its crisp snap. If water is lost, the pressure decreases. the cells shrink slightly. The leaf becomes soft and droops. Wilting is simply the loss of turgor pressure. This is why placing wilted lettuce in cold water can sometimes restore firmness. Water moves back into the cells through osmosis. The vacuoles refill. Pressure returns. The leaf regains some of its structure. Lettuce grows best in cool conditions. It is considered a cool season crop. High heat can cause lettuce to bolt, meaning it sends up a tall flowering stalk prematurely. When bolting occurs, leaves often become more bitter. This bitterness comes from compounds that the plant produces as it shifts toward reproduction. Different types of lettuce show how selection has shaped structure. Some varieties, like romaine, form elongated heads with sturdy ribs. Others, like butterhead lettuce, form soft, rounded clusters. Iceberg lettuce develops a tightly packed head with crisp, pale leaves. These forms all come from the same species, but reflect different patterns of leaf arrangement and density. If you slice through a head of lettuce, you can see how leaves overlap. Each leaf grows from a central stem, wrapping around those inside it. In head-forming varieties, this overlapping becomes more compact over time. The inner leaves remain protected from direct sunlight and often appear lighter in color. The green color in lettuce leaves comes from chlorophyll. Chlorophyll allows the plant to capture sunlight and convert it into chemical energy. In darker green varieties, chlorophyll concentration is higher. These leaves may also contain additional pigments and nutrients. Lettuce has a relatively shallow root system. Because its roots do not extend deeply into the soil, consistent moisture is important. If the soil dries out, the plant may struggle to maintain water balance, and leaves can wilt even while still attached to the stem. In a cool field during spring or early autumn, rows of lettuce sit low to the ground. Leaves spread outward in layers. Morning dew may rest lightly on the surface. The plant grows quickly under moderate temperatures, expanding leaf by leaf. Unlike root vegetables that hide their energy underground, lettuce displays its structure openly. It is a plant built for rapid growth rather than long-term storage. Its life cycle can be short. Seeds are planted, leaves form. The plant reaches harvest size within weeks. Its crispness is a balance of water, cell walls, and gentle pressure. Its softness comes when that balance shifts. From this light and water-filled leaf, we begin to deepen into darker greens, where pigments concentrate and the chemistry inside each cell grows more complex. If lettuce is pale and light, spinach is darker and denser. Spinach, Spinacia oleracea, traces its roots to ancient Persia, in a region that is now part of modern-day Iran. From there, it spread eastward into Asia and westward through trade routes into the Mediterranean and Europe. By the Middle Ages, spinach had become a familiar crop in many parts of the world. Unlike head-forming lettuce, spinach grows as a rosette. Its leaves radiate outward from a short central stem near the ground. Each leaf grows quickly, stretching toward light, widening and thickening as it captures energy from the sun. The deep green color of spinach comes from a high concentration of chlorophyll. Chlorophyll is the pigment that allows plants to absorb sunlight, particularly in the blue and red parts of the spectrum. The energy captured by chlorophyll drives photosynthesis, the process by which carbon dioxide and water are converted into sugars. But spinach leaves contain more than chlorophyll. Spinach is especially known for its vitamin K content. Vitamin K plays an important role in blood clotting. It helps activate proteins that allow blood to form clots when needed. Without adequate vitamin K, the clotting process becomes less efficient. Spinach also contains folate, a B vitamin involved in DNA synthesis and cell division. Folate supports the formation of new cells, which is particularly important during periods of growth. Inside each spinach leaf, cells are tightly packed with chloroplasts. Chloroplasts are small structures where photosynthesis takes place. The abundance of chloroplasts contributes to the leaf's rich green color. Spinach leaves are relatively thin, but densely layered with photosynthetic tissue, allowing them to grow quickly in favorable conditions. Spinach prefers cool weather, much like lettuce. It is often planted in early spring or autumn. In hot temperatures, spinach may bolt, sending up a tall flowering stalk. When this happens, the leaves can become more bitter and less tender. Cold tolerance is one of spinach's strengths. The plant can withstand light frosts. In cool conditions, growth may slow, but the leaves remain viable. This ability allows spinach to grow in climates where other leafy greens may struggle. Spinach also contains naturally occurring compounds called oxalates. Oxalates can bind to certain minerals, such as calcium, within the plant tissue. When consumed in very large amounts, oxalates may reduce mineral absorption. However, in moderate quantities as part of a varied diet, spinach remains a nutrient-rich food. The presence of oxalates is part of the plant's internal chemistry. Many plants produce secondary compounds that serve protective roles. In spinach, these compounds coexist with vitamins, minerals, and antioxidants. Leaf growth in spinach can be rapid. Given adequate moisture, moderate temperatures, and sufficient nitrogen in the soil, new leaves unfold steadily from the center. Each leaf begins as a small, folded structure. As cells divide and expand, the leaf flattens and spreads outward. Nitrogen plays a key role in leaf development. Nitrogen is a major component of chlorophyll molecules and amino acids. When soil nitrogen is sufficient, spinach leaves tend to be darker and more vigorous. If nitrogen is limited, growth may slow and leaves may appear lighter in color. If you imagine a spinach field on a cool morning, rows of dark green leaves lie close to the soil. The surface may appear slightly glossy from dew. Each leaf is broad and slightly curved, catching light at different angles. Unlike vegetables that store large amounts of energy underground, spinach invests in quick, above-ground growth. Its life cycle is efficient. It grows, produces leaves, and prepares to flower within a relatively short time frame. Its deep green color reflects concentrated photosynthesis. Its chemistry reflects a balance of nutrients and protective compounds. Its growth reflects adaptation to cooler climates and shorter seasons. From this dark, nutrient-dense leaf, we begin to turn toward another member of the leafy world, one that does not spread outward in open leaves, but instead gathers itself inward, forming a tight and layered head near the ground. Along rocky coastlines of ancient Europe, a hardy green plant once grew low against the wind. Its leaves were thick and slightly waxy. Salt from ocean spray touched its surface. This wild plant, known as Brassica oleracea, would eventually give rise to many familiar vegetables, including cabbage. Cabbage is one expression of that wild coastal species. Unlike spinach, which spreads its leaves outward in an open rosette, cabbage gathers its leaves inward. As the plant grows, new leaves form at the center. Instead of stretching outward freely, they begin to overlap tightly. Each new leaf wraps partially around the previous one. Over time, the center becomes compact and rounded. A head forms. This head is not a single structure, but many leaves pressed together. The formation of the cabbage head depends on steady growth conditions. Cool weather encourages tighter development. As temperatures drop in autumn, the plant continues to photosynthesize but does not rush into flowering. Instead, it invests energy into building that dense cluster of leaves. Like other members of the Brassica family, cabbage is typically a biennial plant. In its first year, it forms leaves and stores energy. If left unharvested into the second year, it sends up a tall flowering stalk. The tight head opens as the central stem elongates. Yellow flowers appear, followed by seed pods. The wild ancestor of cabbage did not form such a compact head. Its leaves were more loosely arranged. Through selective breeding, farmers chose plants with leaves that overlapped more tightly. Over many generations, this trait became stronger. The result is the familiar spherical cabbage head. Cabbage is known for its resilience in cool climates. It can tolerate light frosts and even brief periods of freezing temperatures. Cold weather often enhances sweetness, as the plant converts some starch into sugars as protection. Against frost damage, this conversion helps stabilize cell structures during cold stress. Inside cabbage leaves, cells are layered and structured. The outer leaves tend to be darker green because they are exposed to more light and contain more chlorophyll. Inner leaves, protected from sunlight, are paler. The overlapping structure also reduces moisture loss from the center of the plant. Cabbage contains vitamin C, an antioxidant involved in immune function and tissue repair. Historically, cabbage and other vitamin C-rich vegetables played an important role in preventing deficiency during long winters or sea voyages. The dense head allowed cabbage to be stored for extended periods in cool conditions. When harvested, cabbage can remain firm for weeks if kept cold. Its tightly packed leaves protect the interior from rapid dehydration. The outermost leaves may dry slightly, forming a protective layer. Cabbage can also undergo a gentle transformation through fermentation. When finely sliced and combined with salt, natural bacteria present on the leaves begin to convert sugars into lactic acid. This process lowers the pH and preserves the vegetable. Fermentation is calm and gradual. It does not destroy the leaf structure entirely, but softens it while creating a tangy flavor. This transformation reflects the plant's stored sugars interacting with microscopic life. The Brassica oleracea species has been shaped into many forms beyond cabbage. Broccoli, cauliflower, kale, and Brussels sprouts all come from the same wild ancestor. Each was selected for a different trait: leaves, buds, stems, or compact heads. In cabbage, the trait favored was inward folding. If you imagine a cabbage field in late autumn, you might see rows of rounded green heads resting close to the soil. Their outer leaves spread slightly, but the center remains tight. Beneath the soil, roots anchor the plant firmly. Above, the layered head holds months of captured sunlight within its overlapping leaves. cabbage grows by gathering itself inward, building structure from repetition. Leaf upon leaf, layer upon layer. From this tightly wrapped head of leaves, we begin to move toward another expression of the same plant family, one that shifts focus away from leaves and towards something else entirely. The flower buds that have not yet opened. From the same wild coastal plant that gave us cabbage, another form quietly emerged. Instead of tightening its leaves inward, this version focused its energy upward. Broccoli, Brassica oleracea variety italica, is not grown for its leaves or its roots. It is grown for its flowers, specifically for flower buds that have not yet opened. Broccoli was shaped through selective breeding in the Mediterranean region, especially in Italy, many centuries ago. Farmers noticed that some plants produce larger clusters of flower buds. Instead of allowing the plant to complete its flowering cycle, they harvested these immature buds while they were still compact and green. If left unharvested, the tight green head of broccoli would not remain as it is. The buds would loosen. Yellow petals would unfold. The plant would flower and eventually produce seed pods. What we eat is a pause stage in that process. The broccoli head is made of many small buds attached to short stems. Each bud is a developing flower. Together, they form a dense cluster called an inflorescence. Beneath the main head, side shoots may form after the central head is removed, continuing the plant's growth. Like cabbage, broccoli thrives in cooler weather. Moderate temperatures support the formation of tight buds. Excess heat can cause the buds to loosen prematurely. Consistent moisture and sufficient soil nutrients, especially nitrogen, help maintain vigorous leaf and bud growth. Broccoli contains compounds called glucosinolates. These sulfur-containing compounds are found in many members of the Brassica family. When plant tissue is damaged by chewing, cutting, or crushing, an enzyme called myrosinase converts glucosinolates into other compounds. One of these compounds is sulforaphane. Sulforaphane forms when glucoraphanin, a specific glucosinolate, is transformed by myrosinase. In the plant, these chemical reactions help deter insects and herbivores. The sharp, slightly bitter taste of broccoli is part of its natural defense system. In human nutrition research, sulforaphane has been studied for its potential antioxidant and protective effects within cells. It interacts with pathways involved in cellular stress responses. These processes occur quietly at the molecular level, reflecting the plant's internal chemistry, continuing its work after harvest. The green color of broccoli comes from chlorophyll in the unopened buds and stems. Beneath that green surface, cells are actively dividing. Even at harvest, the buds remain metabolically active. If stored too long at room temperature, the buds may begin to yellow as chlorophyll breaks down and the plant edges closer to flowering. Proper storage slows this process. Cool temperatures reduce respiration rates within plant cells. Like many vegetables, broccoli continues to respire after harvest. Respiration is the process by which stored sugars are used to produce energy. Lower temperatures slow this metabolic activity, preserving freshness. If you look closely at a broccoli head, you can see repeating patterns. Small branching stems divide into smaller branches, which divide again into tiny buds. This pattern reflects the plant's growth habit. Each branch supports a cluster of buds arranged in a structured, almost geometric form. Broccoli shares its species with cabbage, kale, cauliflower, and Brussels sprouts. All are variations of Brassica oleracea, shaped by selection for different plant parts. In broccoli, the focus became the flowering stage, paused before blooming. If you imagine a broccoli field in cool spring light, you might see rows of sturdy green plants, each topped with a compact head. Leaves spread outward around the stem, gathering sunlight. At the center, the bud cluster rests tightly packed. This vegetable is a moment held in suspension. It is a flower not yet opened. It is growth paused at the edge of transformation. From these tightly gathered buds, we move toward another plant that also grows above ground, carrying its chemistry in bright colors and shifting heat, ripening from green to red beneath the sun. In warm sunlight, hanging from slender green stems, peppers slowly change. Peppers belong to the genus Capsicum. Several species were domesticated in the Americas thousands of years ago. Archaeological evidence suggests that people in regions of present-day Mexico and Central and South America were cultivating peppers more than 6,000 years ago. Unlike some vegetables that trace back to a single domestication event, peppers appear to have been domesticated multiple times in different regions. There are both sweet and hot varieties, but they share similar origins. The differences come from chemistry. Peppers begin as small green fruits. Botanically, like tomatoes, they are fruits because they develop from fertilized flowers and contain seeds. In the early stages, chlorophyll gives them their green color. As they ripen, chlorophyll breaks down. Other pigments increase. Red peppers develop pigments called carotenoids, including capsanthin and beta-carotene. Yellow and orange varieties produce different combinations of pigments. These color changes signal that seeds inside the fruit are mature. Ripening also affects flavor. Sugars increase. Acidity shifts. Texture softens slightly. The fruit becomes more attractive to animals that may help disperse the seeds. Hot peppers contain a compound called capsaicin. Capsaicin is concentrated in the white inner membranes that hold the seeds, rather than in the seeds themselves. This compound interacts with receptors in the mouths of mammals, creating a sensation of heat. The heat is not actual temperature, but a chemical signal that activates pain-sensitive pathways. Capsaicin evolved as a defense. Mammals tend to chew seeds, which can destroy them. Birds, however, do not perceive capsaicin in the same way. The receptors that respond to capsaicin in mammals are less sensitive in birds. As a result, birds can eat hot peppers without discomfort and later disperse the seeds intact. This selective defense favors seed survival. Not all peppers contain high levels of capsaicin. Sweet bell peppers have little to none. Through selective breeding, humans have shaped peppers across a wide range of heat levels. The Scoville scale measures capsaicin concentration, reflecting how much of the compound is present. Peppers are also rich in vitamin C. In fact, many ripe peppers contain more vitamin C by weight than citrus fruits. Vitamin C supports collagen formation and acts as an antioxidant within the body. The bright color of ripe peppers often reflects high levels of beneficial plant compounds. Pepper plants prefer warm climates and long growing seasons. They require well-drained soil and consistent sunlight. In cooler regions, Gardeners may start pepper seeds indoors before transplanting them outside once temperatures rise. The structure of a pepper fruit is simple yet precise. The outer wall, called the pericarp, forms the fleshy part we eat. Inside, chambers hold seeds attached to a central structure known as the placenta. In hot peppers, capsaicin is produced in glands along this inner tissue. As peppers ripen, their respiration rate changes. Like other fruits, they continue to respire after harvest. Warm temperatures accelerate ripening and softening. Cooler storage slows these processes. If you imagine a pepper plant in late summer, you might see fruits at multiple stages. Some remain green and firm. Others glow red, orange, or yellow in the sun. Leaves spread outward to capture light, while roots anchor the plant in warm soil. The chemistry of peppers tells a story of adaptation. Pigments change as fruit matures. Capsaicin deters certain animals while sparing others. Sugars rise as seeds prepare for dispersal. Color signals ripeness. Chemistry signals defense. From these bright fruits shaped by heat and light, we begin to look downward again, toward the ground that supports them all. The quiet system beneath the surface where nutrients move unseen and roots form their steady network. Before roots can thicken, before leaves can unfold, before fruits can ripen, there is soil. Soil is not simply dirt. It is a living system made of minerals, organic matter, air, water, and countless microscopic organisms. Beneath every carrot, potato, lettuce leaf, and pepper plant lies a quiet network that supports growth from below. At its simplest level, soil provides anchoring. Roots push downward and outward, holding the plant upright. But anchoring is only the beginning. Soil is also the plant's source of essential nutrients. 3 major nutrients are especially important for vegetable growth: nitrogen, phosphorus, and potassium. Nitrogen supports leafy growth. It is a key component of chlorophyll, the pigment that allows plants to capture sunlight. It is also part of amino acids, which are the building blocks of proteins. When nitrogen is sufficient, leaves tend to be darker green and more vigorous. If nitrogen is limited, leaves may appear pale and growth may slow. Phosphorus plays a central role in root development and energy transfer within cells. It is part of molecules like ATP, which carry energy inside plant cells. Strong root systems depend on adequate phosphorus in the soil. Potassium supports overall plant function. It helps regulate water movement within cells and supports enzyme activity. Potassium contributes to resilience, helping plants manage stress from drought or temperature changes. These nutrients do not exist in isolation. They move through soil in dissolved forms, carried by water. Roots absorb them through tiny root hairs that increase surface area. The interface between root and soil is dynamic. Water flows in. Minerals dissolve and move toward the root surface. Ions pass through cell membranes. Soil texture affects how well this process works. Sandy soils have larger particles and drain quickly. Water moves through them easily, but nutrients may wash away faster. Clay soils have very small particles that hold water tightly. They retain nutrients well, but can become compacted, limiting airflow. Loam soil, which contains a balance of sand, silt, and clay, often provides ideal conditions. It holds moisture, while still allowing air pockets. Those air spaces are important. Roots need oxygen for cellular respiration. Without adequate airflow, roots can suffocate. Beneath the surface, soil is also alive. Microbial life plays a central role in nutrient cycling. Bacteria and fungi break down organic matter, such as fallen leaves and plant residues. As they decompose this material, nutrients are released back into forms that plants can absorb. One particularly important partnership occurs between plant roots and mycorrhizal fungi. These fungi form associations with roots, extending thin filaments into the soil. The fungal network increases the surface area available for nutrient absorption. In exchange for sugars from the plant, the fungi help deliver phosphorus and other minerals. This underground collaboration is quiet and continuous. Soil also stores water. After rainfall or irrigation, water fills spaces between soil particles. Some of this water drains away under gravity. Some remains held by surface tension. Roots draw from this stored water between rain events. The ability of soil to retain moisture influences how often vegetables need irrigation. If you imagine slicing through a section of soil, you might see layers. Darker topsoil rich in organic matter sits near the surface. Below that, lighter subsoil contains more minerals and less organic content. Roots may extend into both layers, depending on the plant species. Healthy soil supports steady growth. It provides nutrients in balance. It drains excess water, but holds enough moisture. It contains life that recycles organic matter and forms symbiotic relationships. All the vegetables we have explored, roots, bulbs, leaves, buds and fruits, depend on this foundation. Without soil structure and nutrient flow, the cycles of growth would slow. Beneath the visible world of stems and leaves, soil carries on its work. It binds particles together. It filters water. It hosts microbial networks that interact with roots. It is a quiet system, largely unseen, yet essential. From this living foundation beneath the surface, we begin to widen our view. Beyond individual nutrients and root interactions, vegetables also grow within larger patterns, seasonal rhythms, planting cycles, and rotations that shape fields year after year. If you stand at the edge of a field at the beginning of a new season, the soil may look still and quiet. Rows are not yet visible. Leaves have not yet unfolded. But even in that stillness, a plan is already forming. Vegetables do not grow the same way in the same soil forever. The land changes with each harvest. Nutrients move. Microorganisms shift. Moisture patterns adjust. To keep soil healthy over time, farmers follow a rhythm known as crop rotation. Crop rotation means planting different kinds of crops in the same field over a sequence of seasons or years. Each vegetable draws nutrients from the soil in its own pattern. Leafy crops like lettuce and spinach use large amounts of nitrogen because nitrogen is a key part of chlorophyll and proteins. Root vegetables such as carrots and potatoes rely heavily on phosphorus to support root development and energy movement within cells. Fruiting plants like tomatoes and peppers depend on balanced levels of potassium to regulate water and support fruit formation. If the same crop is planted in the same place year after year, the nutrients it depends on most may gradually decline. the soil can become unbalanced. Growth may weaken, yields may shrink. Rotation helps prevent this. One of the most important groups in rotation systems is legumes. Legumes include plants such as peas and beans. These plants form partnerships with special bacteria in small nodules along their roots. These bacteria can take nitrogen gas from the air in the soil and convert it into a usable form for plants. This process is called nitrogen fixation. When legumes grow in a field, they contribute nitrogen to the soil system. After harvest, some of this nitrogen remains available for the next crop. By alternating nitrogen-demanding vegetables with nitrogen-restoring legumes, farmers maintain balance. Crop rotation also interrupts pest and disease cycles. Many insects and soil pathogens specialize in certain plant families. If cabbage is planted repeatedly in the same field, insects that feed on brassica plants may increase in number. Fungal spores may persist in the soil. By switching to an unrelated crop the following season, the food source for those pests disappears. Without their preferred host, their populations decline naturally. This pattern of disruption reduces the need for chemical intervention. Seasonal timing is part of the cycle as well. Cool season crops grow best in spring and autumn when temperatures are moderate. Warm season crops thrive in summer heat. After harvest, some fields are left fallow, allowing the soil to rest. In other cases, cover crops are planted. Cover crops protect the soil surface from erosion and help maintain organic matter. As cover crops grow, their roots hold soil particles together. When they are later turned back into the soil, they contribute organic material. This organic matter improves soil structure, helping it retain water and maintain airflow. Ancient agricultural systems often relied on rotation without formal measurement. In parts of Europe, farmers practiced a three-field system. One field grew crops in spring, another grew crops in autumn. The third rested. Livestock sometimes grazed on resting fields, returning nutrients through manure. Modern agriculture may use soil testing and detailed planning, but the principle remains consistent. Soil must be managed over time. If you imagine a field across several years, you would not see the same pattern repeated endlessly. One year, leafy greens fill the rows. The next, beans climb upward. After that, root vegetables form beneath the soil. Each crop leaves behind subtle changes in nutrient levels, root structures, and microbial activity. Rotation creates a loop, not a straight line. It is a way of working with the soil's natural limits rather than against them. Above ground, leaves rise and fruits ripen. Below ground, nutrients move and microorganisms respond. The field breathes through cycles of planting and renewal. And beneath this broader pattern of field management, something even more intricate is happening. Roots extending into soil, meeting fungal networks, exchanging sugars and minerals in a partnership too small to see, yet essential for growth. Beneath every vegetable plant, below the leaves and stems that catch the light, there is a quieter world. Roots extend outward and downward into the soil, forming branching networks. At first glance, roots may seem simple. They anchor the plant. They absorb water. But when we look closer, we find a system of exchange and partnership that is steady and precise. At the tip of each root is a region of growth. Cells divide and elongate, allowing the root to push gently through soil particles. Just behind the growing tip are thousands of tiny projections called root hairs. These root hairs are thin extensions of single cells. They greatly increase the surface area available for absorption. Water in the soil carries dissolved minerals. Nitrogen, phosphorus, potassium, calcium, and magnesium move toward root surfaces in solution. Through specialized membrane channels, these nutrients pass into root cells. But roots do not work alone. Many vegetable plants form partnerships with fungi called mycorrhizae. The word mycorrhiza means fungus root. In this partnership, fungal threads called hyphae wrap around or even enter root tissues. These threads extend far beyond the reach of the plant's own root hairs, spreading into tiny spaces between soil particles. The fungal network acts like an extension of the root system. In exchange for sugars produced through photosynthesis, the fungi deliver nutrients, especially phosphorus, to the plant. Phosphorus does not move easily through soil. It can bind tightly to soil particles. The fine hyphae of fungi are able to access these bound nutrients and transport them back to the root. This exchange is balanced. The plant provides carbohydrates. The fungus provides minerals. Below the surface, this cooperation continues quietly, unseen. Once water and nutrients enter the roots, they must travel upward. Inside plant tissues are two main transport systems, xylem and phloem. Xylem carries water and dissolved minerals upward, from the roots to the leaves. This movement is driven largely by transpiration, the process by which water evaporates from tiny openings in leaves, called stomata. As water evaporates, it creates a pulling force that draws more water upward through the xylem tubes. This upward flow is continuous during the day when leaves are open and transpiration is active. Phloem moves in the opposite direction. It carries sugars produced in the leaves downward to other parts of the plant. Through photosynthesis, leaves convert carbon dioxide and water into glucose. Some of that glucose is used immediately. The rest is transported through the phloem to roots, bulbs, tubers, and developing fruits. This system allows energy to move where it is needed. In a carrot, sugars travel down and are stored in the thickening taproot. In a potato, sugars move into developing tubers. In lettuce and spinach, sugars support expanding leaves. In tomatoes and peppers, sugars accumulate in ripening fruits. Roots also respond to gravity through a process called gravitropism. Specialized cells in the root tip contain small particles that settle downward, helping the plant sense direction. As a result, roots grow deeper into the soil, while shoots grow upward toward light. The soil around roots is called the rhizosphere. This narrow zone is rich in microbial activity. Bacteria, fungi, and other microorganisms gather near roots because plants release small amounts of sugars and organic compounds into the surrounding soil. These exudates feed microbial communities. In return, microbes help break down organic matter and release nutrients in forms plants can absorb. If you imagine slicing through a section of soil beneath a vegetable field, you would see not empty space, but a web of roots and fungal threads weaving between particles. Water films cling to soil surfaces. Nutrients dissolve and move in slow currents. Above ground, leaves sway gently in sunlight. Below ground, A network of exchange continues without pause. Roots absorb, fungi extend, water rises through xylem, sugars descend through phloem. This system connects soil to leaf, darkness to light. And from this quiet movement of nutrients within plants, we begin to turn toward how those same nutrients continue their journey once vegetables are harvested, how they interact with the human body in steady and subtle ways. When we harvest a vegetable and bring it into the kitchen, its story does not end. The nutrients stored within its cells continue their journey, moving from soil and sunlight into the human body. Vegetables nourish us in many ways, often quietly and gradually. One of the most important contributions vegetables make is fiber. Fiber is a type of carbohydrate found in plant cell walls. Unlike sugars and starches, fiber is not fully broken down by enzymes in the small intestine. Instead, it passes into the large intestine, where it becomes food for beneficial gut microbes. These microbes ferment certain types of fiber, especially soluble fiber, producing short-chain fatty acids. These fatty acids support the health of the intestinal lining and help maintain balance in the gut environment. Insoluble fiber, on the other hand, adds bulk and supports regular movement through the digestive system. Leafy greens, root vegetables, and peppers all contain varying amounts of fiber. Though fiber itself does not provide direct calories in the same way sugars do, it shapes digestion in steady and supportive ways. Some vegetables also contain pigments that serve dual purposes. In carrots, the orange pigment beta-carotene is stored within root cells. When we eat beta-carotene, our bodies can convert it into vitamin A. This conversion happens primarily in the small intestine and liver. Vitamin A plays an important role in vision, particularly in low-light conditions. It also supports immune function and the maintenance of skin and mucous membranes. Spinach and other dark leafy greens provide vitamin K. Vitamin K is essential for activating proteins involved in blood clotting. Without adequate vitamin K, the clotting process becomes less efficient. Leafy vegetables supply this vitamin in a form that the body can readily use. Cabbage, peppers, and broccoli contain vitamin C. Vitamin C functions as an antioxidant and supports collagen formation, which is important for skin, blood vessels, and connective tissue. In plants, vitamin C helps manage oxidative stress. In humans, it supports similar protective processes at the cellular level. Vegetables also contain a wide range of antioxidants and phytochemicals. Lycopene in tomatoes, sulforaphane in broccoli, and flavonoids in leafy greens all interact with cellular pathways in subtle ways. These compounds often originate as part of the plant's own defense system against environmental stress. When consumed, they continue to function as biologically active molecules. Another important feature of most vegetables is their low glycemic impact. The glycemic impact of a food reflects how quickly it raises blood sugar levels. Because many vegetables contain fiber and relatively low amounts of simple sugars, they tend to produce gradual changes in blood glucose. This steady release of energy contrasts with foods high in refined sugars, which may cause sharper fluctuations. Even starchy vegetables such as potatoes have layered effects. Freshly cooked potatoes contain digestible starch that can raise blood glucose more quickly. However, when potatoes are cooled, some of that starch reorganizes into resistant starch. Resistant starch resists digestion in the small intestine and behaves more like fiber, feeding gut microbes. Vegetables also provide minerals absorbed from soil. Potassium supports fluid balance and nerve signaling. Magnesium plays a role in muscle and enzyme function. Iron in leafy greens contributes to oxygen transport in the blood, though plant-based iron is absorbed differently than iron from animal sources. When we eat vegetables, we are consuming structures built from sunlight. Sugars created through photosynthesis are stored in roots, stems, leaves, and fruits. Minerals drawn from soil become part of plant tissue. Water held within cells contributes to hydration. Inside the body, digestion begins to break these tissues apart. Cell walls soften. Nutrients dissolve and enter circulation. What began as soil nutrients and light energy becomes part of human metabolism. The exchange is continuous. Plants absorb carbon dioxide and release oxygen. Humans inhale oxygen and release carbon dioxide. Plants store nutrients in physical form. Humans absorb and use those nutrients to sustain movement and thought. And even after vegetables are harvested, their cells remain active for a time. Respiration continues. Water slowly evaporates. Enzymes carry out quiet chemical changes. From nourishment within the body, we turn next to the life of vegetables after harvest. Their continued metabolism, their response to cooling, and the ways we preserve their freshness before they reach our plates. When a vegetable is harvested, it does not immediately become inactive. Even after being removed from the soil, its cells continue to function. They continue to respire. They continue to use stored energy. In many ways, a harvested vegetable is still alive for a time. Respiration is the process by which cells convert stored sugars into usable energy. Inside plant cells, glucose combines with oxygen to produce energy, carbon dioxide, and water. This process happens continuously, both before and after harvest. Once a vegetable is separated from its roots and leaves, it can no longer replace the sugars it consumes. Photosynthesis stops if leaves are removed, or if the vegetable is placed in darkness. But respiration continues, slowly using stored carbohydrates. The rate of respiration affects freshness. Some vegetables, such as broccoli and spinach, have relatively high respiration rates. Their cells use energy more quickly, which means they can lose quality faster, if not cooled promptly. Others, such as onions and potatoes, have lower respiration rates and can be stored for longer periods under the right conditions. Temperature plays a central role in this process. Cooling slows metabolism. When vegetables are stored at lower temperatures, the enzymes involved in respiration work more slowly. This reduces the rate at which sugars are consumed and helps preserve texture and flavor. Refrigeration does not stop biological activity entirely, but it slows it. Water balance also continues to matter after harvest. Vegetable cells contain vacuoles filled with water. As time passes, water gradually evaporates from the surface, especially if humidity is low. As water is lost, turgor pressure decreases. Leaves wilt. Roots become limp. Crisp textures soften. This is why many vegetables are stored in environments with controlled humidity. High humidity reduces water loss. Proper airflow prevents excess moisture from causing decay. Different vegetables require different storage conditions. Leafy greens, such as lettuce and spinach, prefer cool, humid environments. Root vegetables such as carrots can tolerate slightly cooler temperatures and moderate humidity. Onions and garlic require drier conditions to prevent sprouting or mold growth. Potatoes are often stored in cool, dark places to prevent greening and premature sprouting. Light exposure can trigger changes even after harvest. In potatoes, exposure to light stimulates the production of chlorophyll, causing the skin to turn green. Alongside chlorophyll, the tuber may produce higher levels of glycoalkaloids, which can taste bitter. Darkness helps prevent this reaction. Ripening enzymes remain active in fruits treated as vegetables, such as tomatoes and peppers. Even after picking, these fruits can continue to ripen. Enzymes break down chlorophyll and build pigments like lycopene and carotenoids. Cell walls soften slightly as pectin structures change. Sugars and acids shift in balance. This is why a green tomato left at room temperature may gradually turn red. If you imagine a freshly harvested field, baskets of vegetables are gathered and carried away. Though separated from soil, the vegetables are not inert. Inside each carrot, potato, pepper, or cabbage leaf, cells continue their quiet work. Carbon dioxide is released slowly into the air. Water moves within tissues. Enzymes guide subtle chemical changes. Over time, energy reserves decrease. Textures shift. Colors may deepen or fade. Storage extends this period of vitality. Cooling slows respiration. Proper humidity maintains structure. Gentle handling prevents damage that could accelerate decay. Eventually, of course, cellular activity declines. Energy stores are depleted. Microorganisms may begin to break down tissue. But for days or even months, depending on the vegetable, life continues at a slower pace. From harvest to table, vegetables remain part of an ongoing biological process. And beyond individual storage and transport, vegetables exist within a larger system. Grown across continents, cultivated in open fields and greenhouses, shaped by modern breeding techniques, and connected through global networks of exchange. If you step back and look at the world from above, fields stretch across continents. In some regions, greenhouses glow softly at night. In others, open farmland follows the curve of rivers and plains. Vegetables are grown in nearly every climate, where soil, water, and light allow them to take root. Today, China is the world's largest producer of vegetables. Vast agricultural regions supply leafy greens, root crops, peppers, and more to both local and global markets. Across Europe, the Americas, Africa, and Asia, vegetables move from farms to cities in steady streams. Some are grown in open fields under sunlight. Others are cultivated inside greenhouses, where temperature and humidity are carefully controlled. Greenhouse production allows vegetables to grow beyond their natural seasons. Inside these structures, light can be extended, frost can be avoided, and water can be delivered with precision. Plants still follow the same biological processes: photosynthesis, nutrient uptake, respiration, but the environment is shaped more carefully. Modern breeding has also shaped vegetables in new ways. For centuries, selective breeding followed the slow loop of observation and seed saving. Farmers chose plants with desired traits, size, flavor, color, or resilience, and planted their seeds again. In recent decades, hybrid breeding has become common. A hybrid vegetable is created by crossing two carefully selected parent plants. The resulting offspring may combine desirable traits from both parents, such as disease resistance and consistent size. Hybrid seeds often produce uniform crops, which can simplify harvesting and transport. Selective breeding works within the natural variation of a species. Genetic modification, often called GMO, introduces specific genes into a plant's genome to create a targeted trait. Some genetically modified crops are designed to resist pests or tolerate certain conditions. These methods are guided by scientific understanding of plant genetics. More recently, gene editing techniques such as CRISPR have allowed scientists to make precise changes within a plant's existing DNA. Rather than introducing genes from another species, gene editing can adjust sequences already present. These changes may influence traits such as disease resistance or growth patterns. Despite these modern techniques, the foundation remains the same. Vegetables still depend on sunlight. They still rely on soil nutrients and water. They still follow cycles of growth, flowering, and seed formation. Across the globe, vegetables support daily life in ways that are easy to overlook. They grow quietly in fields and gardens. They fill markets and kitchens. They carry nutrients from soil to human bodies. Imagine, for a moment, a world without vegetables. Fields would still exist, but they would be missing the layered greens of lettuce and spinach. The soil would no longer hide carrots and potatoes thickening beneath the surface. Flower buds, like broccoli, would never pause before blooming. Peppers would not shift from green to red in the sun. Onions and garlic would not form their layered bulbs underground. Without vegetables, the cycle of planting and harvest would feel incomplete. The exchange between soil microbes and roots would continue for wild plants, but the partnership between cultivation and nourishment would be diminished. Fiber would be scarce. Many vitamins and protective plant compounds would be harder to find. The quiet balance of nutrients moving from leaf to body would change. Vegetables are sunlight made solid. They are minerals shaped into structure. They are water held in delicate cell walls. They are color created by pigments responding to light. From wild ancestors on rocky coastlines and mountain slopes to modern greenhouses and global markets, vegetables follow steady patterns. Roots descend, leaves unfold, buds gather, fruits ripen, harvest comes, storage follows, seeds are saved. The cycle begins again, and beneath it all, soil holds its quiet network. Fungi extend their threads, water moves through xylem, sugars travel through phloem. Sunlight becomes tissue. Tissue becomes nourishment. Life with vegetables is woven into daily rhythm. Without them, the landscape would feel thinner, less layered, less connected to the slow transformation of light into food. So we return to the beginning. Sunlight touching leaves, roots meeting soil, seeds resting before growth. A quiet system, repeating season after season, steady and patient, shaping fields and bodies alike.